Hello, everyone. Thank you so much for giving me the opportunity to present my ISN skill share. And thank you so much for tuning in to my talk. If you were part of the previous ISN skill share, I believe it is a wonderful platform which gives the expert an opportunity to share their skills with a wider audience. And today is my turn. So I'll be sharing my skills, and my topic of presentation today is cracking the cancer code. And as you can imagine from my presentation, I'll be sharing my skill relevant to my role as a cancer researcher. And I'm very excited to do that. And in the next half an hour or so, we will be discussing some of the core skills which I usually employ on day-to-day basis. And believe me, I'll be making this presentation very engaging. I'll try to do that at least, and relevant to you guys so you can follow on. And if you would like to pursue this area as your career, then it will also give you some of the gist of what this entails. Yes, so let's start. So just a bit of background about myself. So my first degree is in medicine from Dar University of Health Sciences, and I have also practiced as a clinician for about 2 years. So during my clinical practice, I developed this interest in research and to apply creative approaches to come up with a novel diagnostic and novel therapeutic approaches. So this motivated me to take on my master's from University of Bradford in biomedical science. I then went on to do my PhD from the John Vange's Cancer Research Center at Nottingham Trent University in epigenetics, which we will be discussing a bit in my talk today. And then I'm currently part of the Cancer Research UK Cambridge Institute, which is affiliated with University of Cambridge, and currently working on a project related to circulating tumor DNA. We will be discussing that in a bit. So today, as I have said in the beginning, the main focus of my talk will be to discuss the skills which are employed by the cancer researcher in particular and the biomedical scientist in general on day-to-day basis. And of course, at the same time, making this very relevant to you so you can kind of get a feel of how the research is progressing in general, what are motivating different researchers, and what are different approaches which are being taken. So there will be four basic aim of my talk. Firstly, I'll be discussing my skill as a research scientist and daily routine of a research scientist. And then I will be discussing some of the cutting edge researches being taking place in the field of cancer diagnostic. Then we will be putting on our lab coat or a thinking hat, and I have tried to incorporate some game. I usually do these sessions face to face, but in this pandemic, things are a bit different, but we will cope it together. And then, of course, I'll be ending my talk with what the future holds and what promises is offered. So before starting, let me give you some quick disclaimer. The first disclaimer is since I will be discussing this field, different technologies from a very basic point of view, and my whole idea is to give you a gist and a field of those technologies. So I might be missing on some of the area or some of the aspect of those technologies. So if you are interested in some of them, I did include the link in the presentation. So feel free to go and visit them and have a more detailed kind of an overview about them. And of course, I'll be addressing this presentation from a cancer research scientist point of view rather than a clinician point of view. So my more focus will be on how researchers are facilitating and improving cancer diagnostic and cancer management. Okay, so the first question now is how we scientists progress research. So the core and the fundamental basis of a research is a question which you try to answer. So there is definitely a question which the research surrounds. So you have a question, it could be any sort of a question. We will be having a look at some of the example in the next slide. And then to address that question, you design an experiment to find out more about that question, to answer that question. And then you employ alternative approaches to validate the results and to see whether you can substantiate the finding and whether the finding holds true by different methods. So let's suppose if you are a basic scientist and you would like to see how the cells communicate. The cells in our body are the basic units of life, and they are the individual kind of a unit which function, and they are about like millions and millions of cells in our body. So if you're a basic scientist and if you would like to see how the cells communicate, this will be your question. And then in order to answer that question, you will design an experiment. You will identify different molecules that the cells sent to communicate the messages with the different cell. And then of course, you will then validate those molecules that you have identified using different approaches. So it's kind of complete the paradigm. If Let's suppose in another example, if you are running a clinical trial in which you are checking the efficacy of a drug, so you have two groups, the cases and control, you administer your test drug to the case group, and then you compare it with a control which either receives any appropriate control drug or a placebo, and then you see how your drug perform against the control, and then you of course validate it with the different parameters of clinical trial. And these are very relevant in the current circumstances of the pandemic, in which there was then a question how we can manage this COVID infection. And then to address those questions, different approaches were being took. Some of the research group designed antiviral drug, other designed the vaccine, and then both were then put through a rigorous clinical trial. And we can see that how effective and how quickly it was done, and how the vaccine rollout was implemented at such an amazing and such a brilliant and extraordinary pace, which kinds of highlight the importance of biomedical science and the research sector, that how it could change some of the bigger problems which are faced by our society. So these research questions are not only relevant today, they are also relevant in the past. And in fact, past discoveries were kind of driven by those questions. If you see example of Alexander Fleming of the discovery of penicillin. So he discovered penicillin kind of by an accident. So he was working in a lab growing bacteria on these culture plates, and he was going like there are different kind of anecdotes to it, but the fungus grew because he left his plate unattended, and the area around the fungus didn't had any bacteria. So the question was why it didn't have any bacteria. So then further subsequent experiments showed the fungus released penicillin, which has antibacterial property. It kind of given rise to the era of antibiotic. Then if we see the discovery of insulin by Banting, the Frederick Banting, who co-founded insulin, and how it changed the life and the quality of life in the current time. And of course, if you're aware of the Human Genome Project, which was a project implemented about 2 decades ago in 2000, the main idea of the Human Genome Project was to sequence the entire DNA in our human genome. And we will be talking more about this in the next subsequent slides, that why it is important to sequence the basis of DNA in our genome, and how it can facilitate and aid in cancer diagnostic and management, and which is kind of more in line with my area, what kind of work I do at the CRUK. Okay, so whenever you get a chance, do visit this Wellcome Institute Museum in London. So this Wellcome Institute had a library of the genetic code of humans. So it had the DNA sequences which are present in the human genome. So there are about 22 volumes which correspond to 22 pairs of chromosomes. There are 23 pairs of chromosomes in human. 22 are these autosomes, and then there are a pair of a sex chromosome. So if you open one of these books, you can see these letters, and these books are from the top to bottom, cover to cover. Every page are full of these four letters, which correspond to the code or the sequence of the DNA. And these four letters include adenine, thymine, guanine, and cytosine. And the combination of these letter kinds of describe the code which are embedded in our genome, and the cells in our body use these codes to make protein and bring about different functions. So these codes are organized in a double helical structure, which we call the DNA. And currently I was reading one of the news article published earlier in the June, which claims that the human genome is now finally completed. So back in 2000, when the Human Genome Project started, some area of the genome was still not sequenced, especially in the center of the chromosomes, which are called centromeres. So now these areas are also sequenced. And surprisingly, if you just count those four alphabet letter in our human genome, there are about 3 billions of them. And let me give you an interesting fact about just 1 to 2% of all the human genome, about 1 to 2% of those 3 billion bases is relevant to make protein. The rest of them is currently under study. Some of them regulate different area of the genes. So what are the genes? The genes are just the stretches of these DNA codes which bring about a specialized function, either to produce protein or to regulate another gene. So putting it into context of my current work at the CRUK, so the group I'm working with, my group is very interested to see the DNA which are derived from the tumor. So we are working with the circulating tumor DNA. So if there is a tumor in the patient, so these tumor by different mechanism or cell turnover release the DNA into the body, and these cancer-specific tumor DNA are then detected, which can provide valuable information about cancer staging, cancer diagnostic, and also how the patient is responding to the treatment, and if the cancer is growing or it is shrinking. So I'm working with the biological properties of these circulating tumor DNA, how they are different from the normal DNA, and what specific mutation or the base pair changes are contained within them, and how we can use them to provide different information relevant to clinics. So in my area as a cancer researcher or in biomedical sciences in general, the first thing is to plan an experiment. So the planning of experiment, for the planning of experiment, there should be a research question. As I have mentioned before, you need a research question, and then you design an experiment to answer that research question. So let's suppose the current project I'm working on is related to the different biomarkers and circulating tumor DNA. So I'm the research question is, what are different biological property of the DNA between the DNA derived from the normal patient or the normal tissue, or the DNA derived from the cancer tissue. And of course, while designing an experiment, there is a whole load of ethical regulation and a regulatory approval that needed to be taken and needed to be sought. And it is just to make sure that the creative idea and approaches that are being employed are under some regulation or anybody is keeping an eye on them. So after getting the relevant approvals, we collect the samples from patients, and then after that, we process them in the lab. It's definitely not these, like the labs are not fuming with gases and stuff. So we mostly work with DNA sequencing. So we sequence those DNA derived from the blood. So this idea of circulating tumor DNA is very relevant in the terms of liquid biopsy. So you can actually take the patient blood sample and then detect those tumor-derived DNA in the blood. And after designing an experiment and getting the results, we publish them in the journal articles or present them at the conferences, which are then adapted into the clinics. But the process is not that simple. It takes kind of years for a research to translate from the bench side to the bedside. So here I have shown the pathway for drug discovery process. So the drug discovery pathway is it's kind of a different area, but it kind of give you a gist of how a bench side work is then translated into the bedside. So for discovery of drug, you start with like thousands of compounds, and you do target identification and validation. You then test those compounds in a preclinical models. So you narrow it down to some compound which shows positive result, and then you validate it using the cells which are grown in the lab and different animal models. You then adapt them to clinical trials, and there are different phases of clinical trials which recruits patients, different healthy and cancer patients, and the increasing number of cohorts to make the experiment very rigorous. And as you can see, it could take years for the clinical trials to complete, because we want to follow the patient for a particular amount or particular number of months to see whether they are responding to the drug or if they are developing side effect. And after all those hassles, the research or drug can finally enter into the market, subject to regulatory approval, and then it can be adapted to clinic. But the length of the time taken during the whole process is just to make sure that the compound which is discovered and which is being produced is safe and is effective for use in humans. Okay, so now progressing to the second part of my talk, in which I will be discussing some cutting edge researches which are taking place in the field of cancer diagnostic. So one of the important area which is currently being explored is the live cell imaging. Currently the scientists are imaging live the cells and how the cells are interacting. But it is particularly relevant in the field of immunotherapy, where the scientists would like to see how the immune cells are killing or are kind of combating cancer cell or different pathogens. So this is a video in which you can see the T cells killing the cancer cells. So I'll play the video for you. So here you can see these blobs of cancer cells which are being attacked by the immune cell. They do them naturally, but in the cancer condition, cancer cells kind of find a way to evade them. So there is a booming area of immunotherapy in which those immune cells are trained to identify cancer cell and then kill them. Isn't it exciting? Oops. Yeah, apart from that, the sequencing technology has improved significantly. So the picture you see above here on the left is the sequencing which were employed at the time of Human Genome Project. So there were like factories of these machines which were sequencing the human genome, and it took like about 3 to 4 years to sequence the entire genome. But now with that, single machine has replaced the entire factory, and now you can sequence the human genome in mere like a couple of days. Isn't that like how the technology has progressed in the last 2 decades? And with this sequencing, we now know more about the mutation, the base changes, and what actually makes the cell cancerous, and then how it could be targeted. So now I have a very interesting activity for you. So in this activity, the DNA sequences are the 4 letters which we discussed previously, the adenine, cytosine, thymine, and guanine. So these are the DNA sequences derived from the normal patient and a patient with a melanoma, a kind of a skin cancer. So what I would like you to do is to identify a mutation. A mutation is like changes in the base pair. For example, this A might not be A here, it might be C or G. So you have 15 seconds to identify mutation. Okay, so time starts now. And believe me, it's not very easy. It's just like the 500th base pair sequence. Okay, time up. So as I was saying, it's just 500 bases, and the human have about 3 billion bases. So if you were to able, like if you were able to identify and spot the mutation, then congratulations, you have a very nice eyesight, but I kind of find it very difficult. So the mutation is here. The T or the thymine in the normal tissue is replaced in the melanoma by adenine. So this single base pair mutation, or we call SNPs, or single nucleotide polymorphism, kinds of increase the potential or increase the activity of one of the protein called BRAF. And this increased activity of the BRAF protein is what that leads to the skin cancer or melanoma. But thankfully, due to this sequencing of the human genome, we now know that these mutations do exist, and there are therapeutic approaches being employed to tackle these mutations. There are compounds available which decreases the activity, the increased activity of the BRAF protein, thereby improving the management of patients with melanoma. And with that, increasing cost of sequence, with that, increasing efficiency of the sequencing and decreasing costs. And now we are able to generate more and more data of the sequencing, of the sequences between normal and the cancer tissue. It kind of gave rise to the field of bioinformatics, in which we need help from the computers to assist us in finding the mutation. And they are just not like single base pair mutation. There are different kinds of mutation, insertion, deletion. So the computer software and tools really help us identify those and kind of predict the outcome of patients. So if you are if this topic excites you, please do have a go with this link. It is a game in which you are competing against the artificial intelligence or the AI model to detect the variants in the deep variants, or these variants in the human genome, and you can see whether you can outsmart the computer. Okay, so now it's time to time to put on our thinking hats and to answer some question about the human biology and see how we know well about our body. Okay, so let's put on our science cap. Okay, so the first question is, which is the smallest bone in the human body? I'll give you 5 seconds to think about it. Which is the smallest bone in the human body? And if your answer is one of these digit bone or the finger bone, I'm sorry, your answer is not correct. It is actually the bone in our middle ear, which is even smaller than the bones in our fingers. Okay, the second question, how many genes in our genome code for a protein? So I'll start the timer, you have 10 seconds. So how many genes in the human code for a protein? Okay, time's up. So there are about 22,000 of the genes that codes for a protein, but or we call them protein coding gene. And interestingly, even banana still share about 60% of the same DNA as human. So we as a human share more than half of our DNA with banana. Isn't that amazing? Okay, so lastly, like let's suppose you're a scientist and you want to test the function of a gene. What or which approaches you could use? So I assume you might not know about some of these, but still have a go, but we will be quickly discussing about them. Yeah, time up. So the answer is all of the above. So if you would like to check the function of a gene, you can use the CRISPR-Cas9 system. So the CRISPR was a system which was designed by an inventor a couple of years ago, and they got the Nobel Prize in chemistry, I guess last year. So using the CRISPR, we can actually edit the genome. We can remove the segment of a gene. So if you would like to see a function of a gene, you can remove that gene from the genome and see what effect it causes. You can also use gene silencing, in which you silent the function, you silent the function of the gene, and the pre-recombinase, in which you turn the gene on and off as you will, and you then check the function which are impacted by turning the gene on and turning the gene off. So these are the various ways we kind of discover the function of the human gene. Okay, so let's move forward. So a little bit more about CRISPR. So as I was saying, the CRISPR is kind of a copy-paste tool. So using the CRISPR technology, you can actually cut the segment of the segment of the genome you like, and then insert any other segment here, or just to see the function of the gene or the genome or the DNA without that particular gene. And of course, these kind of technologies help us answer so many questions, and it's also being translated into clinic for some of the diseases. But the word of caution is, of course, as I was saying, that we do need ethical and regulatory approval. So a few years ago, there was this scandal of about a CRISPR baby in which the scientists has removed, a particular scientist has removed a gene using CRISPR from a baby, from a human baby. And we don't exactly know what's the function of that gene. We do know broadly, but then it was not exactly known. And that's why it kind of highlight that the need of an ethical regulation. So any experiment that is planned should be regulated by like a regulating body. So lastly, I'll be sharing what the future holds. There's definitely light at the end of the tunnel, and the way I see the field is progressing. It upholds so many promises and so many potential. So what I think that in the future, the medicine will be personalized due to the decreasing cost and increasing speed of sequencing. So every one of us would have their genome sequence. In contrast to the old saying of, one drug fit all model, we will now shift more to the drug for a particular individual at a particular time kind of a thing. So the drug will be more kind of following precision and personalized medicine, depending on your genetic makeup and the way your body function, the way your genes expresses. The treatment modality could be tailored from one patient to another. like not like it could be tailored according to the specific patient. And of course, what I'm also seeing in the future is the booming of the artificial intelligence in bioinformatics. This is the area my current group is working at the CRUK as well. It is coming up with the artificial intelligence or the computer learning models to predict the outcome or to predict how the different mutation and the predict of how the different mutation predict the risk of developing particular condition or particular cancer, and how these all fits in and how well and how good response we can get using a particular treatment. So there are different algorithms being designed. So some of the example are of machine learning and the biology include molecular evolution, protein structure analysis, system biology, and genomics for disease research. So we are using the AI in our current work, in which we have kind of developed a pipeline called ENVAR, which detect patient cancer specific mutation in the circulating tumor DNA. And lastly, I'll conclude my slide discussing some of the invention of 2021. So it kind of tells us how the field is progressing. So now there is like the pocket-based ultrasound devices are available. There's also the virtual reality headset, which could be employed in clinics for rehabilitation. So if a patient has a motor neuron disease, using these augmented reality headset facilitate speedy recovery. And with the booming of the technology and with the uprise of how the technology take part in our daily routine, like smartwatches, smartphone, we kind of have big data from each individual, incorporating different parameters like blood pressure, heartbeat from smartwatches, and of course, if we incorporate their genomic information. So these all kind of big data then fits into how a particular individual will be responding to a treatment, or how likely he's to develop a particular disease or to respond to a particular treatment. But at the same time, there should be some form of regulation and the consent surrounding it. So currently the drones are being employed to transfer life-saving drug and blood across different sites of hospital. And with the increasing use of technologies, the boundaries between different nations are kind of becoming blurred. So the scientists from different regions are collaborating, and now the cohorts that are used in clinical trial are not just limited to the West. We have like a different ethnic cohorts which give us a broader picture, and definitely the personalized medicine and the area like the one I'm working on, the circulating tumor DNA, is showing very promising results. So just concluded, I hope I have given you a glimpse about my field and made you a bit excited. So the science, to conclude, is growing at an unprecedented pace. So by digging deeper, we are finding more question than answer. So the approaches which we designed to answer a question, in fact, raise more question, which is kind of good that we are learning more deeper about that area. And of course, this all be done in a regulated kind of a framework. So thank you so much for tuning in and listening to my talk. I hope you have enjoyed it. If you would like to reach out, feel free to contact me through LinkedIn. This is my LinkedIn ID. And I'm also kind of working on my YouTube channel, making the science simpler for general audience. So do subscribe to my channel. And thank you so much once again.